Okay, so it looks like I'm by myself. I'm just gonna do a quick review for you guys, just to kind of give you a heads up of what you're gonna see on the final exam. So I'll begin with chapter 14. Hey, Destiny, can you hear me?
Yeah, I can hear you.
Perfect. So I was just, no one was there, and so I just decided that I was going to go ahead and go through each of the chapters. But if you have a specific questions or any questions, just let me know.
Okay.
So I'm going to start from chapter 14 and just sort of talk about the things that I expect you guys to know for the exam. So you should know what the blending theory of inheritance is. Remember, like when I highlight stuff in orange, that's a definition you should know. So blending theory simply means that when two things breed together, they're blended together, kind of like a dark purple flower and a white flower would make a light purple flower. And Gregor Mendel sort of looked at this and said, I don't think that that's true. And so he started looking at the pea plants. So you don't need to know dates, but you need to know people. You should know who Gregor Mendel is. He's considered the father of genetics, and so he's kind of an important figure. I don't care if you know any of this stuff, where he's from, or any of those things. It's just kind of telling a story. You should know that he lived in a monastery, and that's where he grew his pea plants in the garden there. And he did these breeding experiments. And you should know why he picked pea plants. Remember, pea plants have male and female parts, and they're covered. And so pollen can't fly around in the air. The plants are self-pollinating. And so if you don't do anything, they're basically making clones of themselves because they're only contributing their own genetics, the male and female parts, the pollen and the stamen. Anyway, but you can cross-pollinate it if you physically open the flower itself and do that. So that's why Mendel picked pea plants, because they're self-fertilizing. You should know the definition of true breeding. So true breeding means that if you bred a purple flower plant with another purple flower plant, it should have purple flowers, the same color as the parents. And then if you bred those brothers and sisters together, they would have the same color as the parents, and so on. So it's kind of like a thoroughbred horse. It means that the genetics are pure. It's just like what you would expect if you went to the store and you bought a packet of seeds and they had a picture of yellow flowers on it. You would expect to plant all those seeds and they would make yellow flowers. And if you let them fertilize each other, they should also, all of their seeds should have yellow flowers, and so on and so forth. So true breeding just means it's homozygous, right? And it could be big A, or it could be little a, little a. And, I'm just using these variables because A is easy, but we use the capital letter for whatever it is. So in this case, purple is dominant, so purple would be big P. White is recessive, so that would be little p. And that's true breeding. All right, so you should know that Mendel crossed purple and white flowers, and and that generation is the parental. So this is basically what he did. He took the parental generation. You should know how to write this out on the test. So because this is how geneticists write this out. And so he took a purple flower plant that we would call big P, and he crossed it to a white flower true breeding plant we would call little p. This is the phenotype, which is what we see. And this is the genotype. This is the phenotype. And so that cross would present, this parent would only be able to donate big P, regardless of which one it was. This parent would only be able to donate a gamete that was little p. So this would be the F1 generation. They would all be big P, little p, all of them, every single one, and they would all be purple, right? And then we let those self-fertilize, because remember, pea plants, they do that if you don't mess with them. And so the genotypes, because all the F1 generation would be big P, little p, that's all they could breed to. And then we could do the Punnett square, but remember that this sperm could fertilize this egg, and so we would get big P, and this one could fertilize this, and we get big P, little p. This one could fertilize this, and we get also big P, little p, and then we would get this fertilize this, and we do little p, little p. And you could do a Punnett square to do this, it'd be the exact same thing. Big P on one and little p on the other, and so you would end up this fertilizes this, big P, this fertilizes that, big P, little p, this is big P, little p, and this is, sorry, little p, little p. Same deal. And so we would get a 1 to 2 to 1 ratio. These together, 3 out of 4 would be purple. So remember the genotype ratio for this cross, which we call a dihybrid cross, you should know that too, is the genotype ratio is going to be 1 to 2 to 1. This is homozygous dominant, heterozygous, homozygous recessive. And the phenotype ratio, I told you guys you should memorize this, for this cross here is 3 to 1 because all of these are purple. So 3/4 is purple and 1/4 is white. So it's going to help you tremendously to know these ratios. I would make flashcards and just memorize them. And a monohybrid, mono is 1, right? So it's only 1 trait. In this case, it's flower color. Hybrid means like a hybrid car. It has gas and electric. So in this case, it has 1 of each allele, a purple and a white. And this is the ratios you get for genotype, 1 to 2 to 1. And remember, you add it all up. So the denominator is 4. And then same thing here. The 3 plus 1 is 4. So the denominator is also 4. So that's what you should know. And it didn't blend, right? So this is where Mendel came up with this idea of these dominant and recessive traits. So he said that the one that disappeared in the F1 generation, in this case here, right, these are all purple, so white's gone. All purple, no white. So he called what disappeared in the F1 generation. So remember, they have the parental generations up here. This is the parents. And they would be true breeding, big P, little p, little p. So for white, since it was gone in F1, he said this is recessive. And this one is dominant. And that's what you need to know, those terms, because that's how geneticists talk. And then remember, in the F2 generation, we have those ratios, right? In this case, we're looking at the phenotypic ratio, which is 3 to 1. Mendel did the math on this, and he counted a whole ton of P plants. We talked about that. This is just an example. You don't need to memorize any of this, but the reason I'm showing you this is to tell you that it's just like flipping a coin. So if you flipped a coin twice and it came up heads twice, that wouldn't be as crazy as if you flipped it 100 times and it came up heads 100 times. So the more that you do a statistical variable, the more likely it is going to come out to what you expect. And in this case, we would expect a 3 to 1 ratio, but if we only counted like 3 individuals, that we might not get that. And that's why they make these giant clinical trials too, with thousands of individuals, just to make sure that their expectation is in the statistical realm of possibilities. I went to graduate school with a girl who did a bunch of crickets, and she was looking at these wasps that were supposed to parasitize them, and all of her crickets died. So she did her scientific study on a single cricket. Like, you can't do that. That's not scientifically valid. You have to do this many times to be able to get this 3-to-1 ratio. All right, so I don't care that you know this, but this is what Mendel called it. We know that these units that you were talking about are genes. And so what Mendel discovered is genes, and that's why he's known as the father of genetics. Remember that the alternative forms of genes are alleles. So the correct definition of a gene would be something that codes for phenotype. So for example, hair color would be a gene, or eye color might be a gene, or bent little finger would be a gene, or widow's peak might be a gene. But there's lots of different variations that we could have. So this one, there's only 2, and this one, there's only 2. But for eye color, you know, I could think of brown and green and blue and you know, hazel or whatever. So there's like, you know, there's several of these, and so these variations, and same thing with hair color. You could probably think of 5 different hair colors. So these are called alleles. And you have 2 alleles for every trait. One you got from mom and one you got from dad. So in the flowers, we would have allele for purple flowers, right? And we would call that big P. And then we would have an allele for white flowers, and we'd call that little p. And because we're dihybrid, and we learned this in chapter 13 and 12, we are diploid, not dihybrid. That means we have 2 chromosomes for every trait. One we got from mom and one we got from dad. That's why we have 2 of these. If you're looking at strawberries, they would have 8 of these because they're octoploid, right? So that's much different. And that's what we mean by alleles. Alleles are alternative forms of a gene. So hair color, an alternative form of that would be red or blonde or brown or so on. The word locus, that we've talked about this before. Locus refers to the location of where that gene is. So the locus for flower color is located here. And remember, these are homologous chromosomes. This could be from mom and this could be from dad. So they're not identical, right? They're homologous. If they were identical, they would be sister chromatids. So make sure you know the word homologous chromosome. We went over this in chapter 12 and 13. All right. So this, I think students have a hard time with genetics because of the math, but you have to be able to do the statistics. A lot of it's pretty easy. You know, a coin has heads and a coin also has tails. And you know the odds of flipping a coin and that coming up heads is 50%. The reason it is because there's 2 outcomes, right? It could either be heads or tails. And so you would just divide one of the outcomes by the total possible outcomes, and each one of these is 1/2, which works out to be 0.5 or 50%. If you're rolling a 6-sided dice, it would be different, right? Because there's 6 outcomes. It could be a 1, a 2, a 3, a 4, a 5, or a 6. So the odds of rolling a 1 on a 6-sided dice is 1 out of 6. And so on and so forth. Now, if you were looking at roulette, and let's say that it came up red and it came up black, right? And we'll just forget that there's a green because that gives the house the advantage. It's just their way of making money, because if you just bet red or black, they would never win because there's a 50% chance it would be red and a 50% chance it would be black. So they would never get any cash off of you. So they throw green in there so that if it ever hits green, they win. And some people bet on green, and that's a sucker bet, but that's a different story. So let's just say red and black is 50-50. Well, every time they spin that roulette wheel, it's an independent event. So you know, they do this at the casino. They have this big sign, And it'll say 17 red, 18 red, 23 red. I don't know the numbers, I'm just making it up, but let's say it said all red, right? And so a lot of people psychologically would be like, okay, it's hit red so many times, it has to hit black. Black is now favored. But the truth is, these are all independent events and they had to be treated independently. So the odds of it coming up black, even though it came up red 1,000 times, is still 50%. And we can use that because they're all independent events. We can multiply those independent events together to give us the probability that two events would occur, right? Like the probability you would trip and also hit your head, right? And so what we're doing here is the probability that you would get either a dominant or a recessive allele and what the outcome of this offspring would be. And this comes down to the law of segregation. So we'll just go, I'm just going to go back and I'll just do this real quick and then we'll move on. But so let's say that we had big P, little p, we're going to do a monohybrid cross. And we know that this is mom, this is dad, and we can do a Punnett square on this. So we know that mom would make a big P egg or a little p egg. Those are only possible eggs she can make. And there's a 50% chance she would make a big P egg and a 50% chance she'd make a little p egg. Same thing with dad for sperm. He could make a big P sperm or a little p sperm. And the odds of that are also 1/2, right? And so the odds of this egg being fertilized by this sperm, well, it's the probability of this being made by this being made. It would be the same thing as tossing a coin and it's saying, what's the odds it's going to come up tails and then tails? Well, it's 50% tails the first time and 50% tails the second time. So we multiply those together to get a fourth. You know, you might ask, well, what's the odds it would come up tails and then heads? Well, it's exactly the same because heads is also 50%. So we can do the same thing here. The odds of us getting a kid like this is 1/4. It's 1/2 times 1/2. The odds of us getting this offspring is also 1/4. This one is also 1/4, and this one is also 1/4 because we're just multiplying the probability of each of these outcomes, just like flipping a coin. And then if it's the same outcome, you can add it together. So we add 1/4 plus 1/4, and we get 2/4, which is 1/2. So we could look at the same thing with dice, right? It's easier for me just to pull up instead of drawing all this out. I'm going to pull up the odds of rolling 2 dice. Okay, so this is good. And this is called a binomial. You don't really need to know that, but let me share this screen. Okay, so here's a binomial, right? We know the probability of rolling. We're rolling 2 dice, right? So the probability of rolling a 1, and hopefully you can see the screen.
It's still showing your slide.
Okay. Okay, can you see it now?
Yeah.
Okay, so the probability of rolling a 1 on a dice is 1 out of 6, which we talked about, right? So the probability of rolling 2 1s, we can use the rule of multiplication, which would be 1/6 times 1/6, which is 1/36, which we see right here. Right, so rolling any of these combinations right here is 1/36. A 1 and a 1, a 2 and a 1, and so on. And every one of these pairs of dice, their probability is 1 out of 36. But you have a much higher odds of rolling a 7 because we can add each of these together, right? So to get a 7, you could roll a 1 and a 6, right? That's 7. The odds of getting a 1 and a 6 are 1 out of 36. But we can add that because you could also roll a 5 and a 2. And so that's 236 or 118, right? We could also roll a 3 and a 4. That's a 7. So then we get 3 out of 36, which reduces to 112. And then we could also get it the other way around. So instead of a 4 on the white dice and a 3 on the black dice, we get a 4 on the black dice and a 3 on a white dice. We have to add that because it's just like big P, or little p, big P. They're the same. It's like rolling a 7. So we add that together and so on. And so you can see that there's actually 1, 2, 3, 4, 5, 6 different ways that you can roll a 7 on the dice. But there's only one way that you can roll a 2 when you're rolling 2 dice. So it's the odds are much higher to roll a 7 than a 2 because we have to add all these probabilities up together. And that's how probability and statistics work. You basically have the rule of multiplication, which is, you know, if I rolled the dice simultaneously, it's no different than me rolling one and then waiting 5 minutes and rolling the other. The probability is still 1/6 that I would hit the number on each of those dice, and I have to multiply those together. You know, if you had a family with 20 males, the odds of you having a female are still 50-50 the next time around. All right, so I'm going to go back to the PowerPoint. And hopefully that explains the rule of addition and the rule of multiplication. So independent events you multiply together. If they're the same event, then you add them together. Okay. So segregation is really simple. We already talked about it. It's if you want to go back and review it, you can look at chapter 13.
It's not showing the slide.
Okay, hang on. Okay. So can you see it now?
Yeah.
All right, perfect. So the law of segregation is really simple. You guys did this whenever you did the lab for mitosis. Remember those tetrads line up on the metaphase plate and prophase 1, and they can line up in either way, right? So in this case, let's say blue is dad. So dad's on the top here for chromosome number 1, and I call it 1 because it's the biggest one. And we'll call this chromosome number 2 because it's smaller. So chromosome number 2 is on the bottom, but dad could easily have both number 1 and 2 on the top. And that is segregation. That means that these things separate during meiosis, and each gamete has an equal opportunity to be produced because of that flip-flopping on the metaphase plate. All right, so these are terms that you're going to have to just memorize if you don't already know it. Homo means same. Zygos is a fertilized egg that we learned in chapter 12 and 13. So homozygous means that you have both of the same alleles, right? So let's say that you're a flower, then homozygous would mean that you're either big P because those are the same, or little p because those are the same. You can't be big P because that's not the same. That's not homozygous. And the reason we refer to as zygous is because everyone starts as a fertilized egg. Everyone starts as a zygote. So whatever that zygote is, every cell that comes from that, from mitosis, is exactly the same genetically. Does that make sense? So that's why we call it homozygous. It's the same in the zygote. We have to distinguish this one from this one. So we call this one homozygous dominant because purple is dominant. And we call this one homozygous recessive because white is recessive. And that's just Mendel's rule. So make sure you know those terms. Make sure you know heterozygous is one of each, right? A phenotype is what you see, so flower color. Genotype is what's in the genes, like big P, little p, little p, big P, little p. Punnett squares are really useful. I use them sometimes just to make sure that I'm doing it right. But if you know the ratios, it's real much. It's more in your favor to know that in a monohybrid cross, it's a 3-to-1 phenotypic ratio. It's a 1-to-2-to-1 genotypic ratio. And so you don't have to do the Punnett square, but hopefully you've done the Punnett square. And if you haven't, you should practice the Punnett square either from the questions in your textbook at the end of chapter 14, or the genetic problems that I put on Canvas. And then we just talked about this. So you could memorize the ratios. It's much easier to do that. And then you don't really need the Punnett square for the smaller ones. You should know what a test cross is. We talked about test cross is when we cross a known to an unknown to find out what this unknown is. So like if I put you out in the field and said, go bring me a purple flower, I might ask you, well, what's its genotype? You wouldn't know unless you did some DNA analysis on it. You couldn't know from looking at it because this and this look the same. The only way to figure that out is to cross it to something we do know. We always know the genotype of the white flowers. They have to be little p, and we see that. Every white flower I picked out of that field would have to be little p, little p. So we cross these, the unknown, right? It could be this, or it could be this, right? And that's a test cross. And we would know from the outcome, these would all be purple. If all the offspring were purple, we would know that the genotype of the unknown is big P, big P. If some were white, and that's actually half, and you could do the Punnett square to test this, then we would know that the unknown is a heterozygous. And that's a test cross. You don't have, these days, most people don't do test crosses because it's easier just to look at the DNA, you know, and you probably figured that out. But, you know, in the 80s, DNA technology didn't really exist. So they really didn't have much options to figure out, you know, what the unknown genotype was. Dihybrid crosses mean that it's di for 2 traits. Hybrid means that they have one of each. So in this case, the parents, this is the example from your book, and it's one of Mendel's crosses he did. So yellow is big Y. Big Y is yellow. That represents the yellow allele. Little y represents the green allele. So remember, we're following the rule of Mendel. It's big Y because yellow is dominant to green. And then big R is round and little r is wrinkled. And again, we're following the rules of Mendel. So it's R because round is dominant to wrinkled. So that's why we use R. So this parent would be a true breeding parent. It's homozygous dominant for both traits, seed shape and seed color. So remember, the genes are seed color and seed shape, and these are the alleles. It can be round or wrinkled. This can be yellow or green. And remember, you have to, you know, think about you. Like if you're having kids, you got to pass on like skin color and eye color. You couldn't just pass on eye color and no skin color. Your kid would be messed up. So you have to pass on at least one of each. And here, the gametes, the only ones that this could pass on if it's doing one of each is big Y and big R. Same thing here, little y and little r. And so we know every single offspring from this true breeding cross is going to be what we call dihybrid, right? It's hybrid for both traits, seed color, seed shape. And we know if we cross those to their siblings, this is very common in genetics, even in mice, right? You have mice siblings, you cross it back to the parent or to their brothers or sisters, and you can know the genetic outcome, kind of like controlling the pollen in Mendel's pea garden. All right, so anyway, you would get this, and just remember, what I want you to know from this is that it's a 9 to 3 to 3 to 1 ratio. Remember, we add these together, so they all add up to 16. So each of this means it's a 16. This is both dominant. These are one dominant and one recessive. This is both recessive. All right. So I would learn the ratio. Otherwise, if you want, I mean, you could write this Punnett square out on the test because you have plenty of time to do it. But if you're taking like the MCAT or something and you have to do this, it's way better to know the ratios because it takes a long time to make these squares, especially if they're complicated like this. And then you should know the shortcuts. So on the test, I might ask you, I might say we bred a dihybrid cat for short tails to a short tail and long fur to a dihybrid cat with short tail and long fur. Short tails dominant and long furs dominant. How many cats would you expect to have long tails and long fur? And you should be able to answer that. I'm not going to do the problem out, but there's examples like that in the problem set. All right, so independent assortment just means that these assort independently. They're not connected together on the same chromosome. We talked about the probability statistics, so that's just a review of that. We did these in class, so make sure you get these are the kind of questions that you'll see on the test, so make sure you know how to do those. Make sure you know the definition of incomplete dominance and complete dominance. Remember, incomplete dominance is in between, where complete dominance is like, it looks exactly like the parent. So in the example we used was snapdragons, there's actually a white pigment and a red pigment, and when they crossbreed, they make pink. So that's incomplete dominance. Whereas in Mendel's world, white would be a default. And so if red was present at all, it would be red. And that would be complete dominance. We talked about codominance, so that means both are expressed. We talked about an AB blood group. So you have the cell and you have the A receptor and you have the B receptor. And since they're both present at the same time, both alleles in the heterozygote are expressed, then that's called codominance. It's super similar to incomplete dominance. And that you just should know an example of codominance is blood types, whereas an example of incomplete dominance is snapdragons. An example of complete dominance is the purple flowers for Mendel. And then multiple alleles, there's lots of examples for this. We talked about multiple alleles for skin color, multiple alleles for blood groups, multiple alleles for hair color, and so on and so forth. So multiple alleles just mean you can have more than 2 forms of that gene, right? Like most of the stuff we talked about before this was, it's either purple or white. It's either red or white. It's either round or wrinkled. It's either yellow or green. So that wouldn't be multiple alleles because it's just 2. So if you have more than 2, like red hair and brown hair and blonde hair, then that's multiple alleles. So blood groups are an easy example of codominance and multiple alleles. Remember the definition of pleiotropy, that just means that multiple phenotypes arise from one genotype defect. Sickle cell is an example of that. I might ask you that on the test. Epistasis is where you get those weird ratios. So if we do a dihybrid cross, we would expect to get a 9 to 3 to 3 to 1 ratio. But in epistasis, that means 2 genes are working together to make the outcome, kind of like a assembly line. And so we talked about, we have the paint, but we also need a painter. And if the painter's not there, then it doesn't matter if we have the paint or not. That causes these ratios to collapse. So you either get a 9:7 ratio or you get a 9:3:4 ratio because these two come together. This is indicating genes in an assembly line. And that's how we figure out these genes in the pathway, like for Roundup resistance or, you know, color in dogs. This also is good for labs, works the same way, Labrador retrievers. All right, so I don't care that you know what epistatic, there's terms called epistatic and hypostatic, and that's something you learn in genetics. But know what epistasis is and know how to identify it. On the test, I might say, I bred up 2 black labs together and I got a 9:7 ratio, or a 9:3:4 ratio of black to brown to yellow. How is that possible? And you would say epistasis. Polygenic, right? Multiple genes are involved in one outcome. So here, not only are these multiple alleles because they're A, B, well, actually, they're not. They're because it's AA, but polygenic means there's multiple genes involved in a single phenotype. Skin color in humans is controlled by at least 3 genes that we know of, probably more. Height is controlled by 21 genes. Your eye color is controlled by at least 3 different genes. Hair color is controlled by 2 genes, and so on. So that's what we mean by polygenic inheritance. Many genes work together to have the phenotypic outcome. A lot of you guys did pedigrees for extra credit. I told some of the students that I would take extra credit all the way up to the end. And that's because I want you guys to learn this. So if you didn't do the extra credit for exam 3, you can still do it. But if you don't follow the rules of the pedigree, you're not going to get credit for it. Squares are males, circles are females. Filled in means that you have that condition or trait. And then we talked about widow's peak and earlobe. I don't care that you know that. You don't need to memorize this. But you should know the rules of the pedigree. So if I put a pedigree on there, you should be able to tell me, yeah, that's a female because it's a round, and that's a male because it's square. You should be able to tell me what a dot in the center means. I might say, is that a carrier? Does that person have it? You should, if it's filled in, you should be able to tell me, yeah, they have it. And then I gave you the extra credit assignment for the Blue People of Kentucky. So if you forgot that, you can go back and look at that lecture. All right, any questions about chapter 14? So before I go on to 16, I'm just going to share this. Okay, so if you go to the home screen and then scroll down in Canvas, underneath the study guides, you'll see the genetics problem sets. So I strongly suggest that you guys work through these problem sets and make sure that you understand how to do these. So I'm going to have office hours today and tomorrow. So if you don't know how to answer these, I can help you during office hours to go over it. But you can also use the answer key to check your answers, which is right here. And so I think I did a pretty good job of explaining this and writing it out. But if you're confused about how I got these answers, then you can come to office hours and I'll help you work through these problems. All right. So make sure you do that, and that'll prepare you for chapter 14. Any questions at all? So I'm going to go on to chapter 16. Okay, so chapter 16 is about DNA replication. So for this, you should know who Frederick Griffith is and what his experiment is. I'm going to ask you that. I'll just tell you straight up. I'm going to say, what are R cells? What would you expect to happen if you got R cells? What are S cells? What would happen if you got S cells? What happened when Griffith mixed living R cells with dead S cells? So you should sort of know this experiment because it's a big deal. You should know the difference between R and S strains. You should know the word transforming. That's an important word in biology and genetics. And like I said, you should know the person, not the date. I don't care about the date. You should also know the Hershey and Chase experiment. This is where they labeled the virus with radioactive material. You should know that proteins only contain sulfur and DNA only contains phosphorus. So that's how they're able to pull this off. And what they figured out is that the DNA ended up inside the bacteria and was radioactive from the radioactive phosphorus. And that's how they discovered that DNA was the genetic material, by using radioactive phosphorus. This I'm probably not going to ask you much about. It's just showing you that this is proof that DNA was the genetic material. You should know who Chargaff is, and you should know what his rule is. So if I ask you on the test, the percent A is 12%. What's the percent G? You should be able to do that problem. No problem. Because of Chargaff's rule. Remember, percent A always equals percent T, and percent G always equals percent C. And you guys did that in the last lab. Whenever you did the gene expression lab, you had to do that, match the A's and the G's and the C's and the T's. You should know who Rosalind Franklin is, that she did the X-ray crystallography, and you should know who Watson and Crick are. So Chargaff, remember, he has the rule that A's equal T's and G's equal C's. Rosalind Franklin figured out the X-ray crystallography of this photo 51 and knew that DNA was a double helix. And then Watson and Crick actually did the model that Franklin helped him with, and therefore they all won the Nobel Prize for it. You don't need to know anything about this, except you should know that DNA is anti-parallel. That means that this end is the 5' end. Remember, 1, 2, 3, 4, 5, and same thing here, 1, 2, 3. So this end is the 3' end. And if that's 5, this is 3. And to prove it, you can do the numbers. And if that's 3, then this has to be 5. And so DNA is anti-parallel. And this, you can see this is upside down from this one. At least the sugars are. You should know DNA is double-stranded. You should know that the way that DNA replicates is the semi-conservative model. We talked about that in lecture. You should know Meselson and Stahl are the ones that showed that, and they did it using heavy nitrogen. You should know the sequence of events for DNA replication. So I'll go over that real briefly. We have an origin of replication. It's A and T rich because there's only 2 hydrogen bonds. G's and C's have 3. That origin of replication is found by an enzyme called helicase. Helicase opens the ORI, the origin of replication, and creates a replication bubble and 2 forks. One fork is on the left side and one fork is on the right side. So it looks kind of like this. So the second thing is that the single-stranded binding proteins bind and keep the bubble open so that the rest of the stuff can do its work. After that, primase has to lay down a primer. I might ask you what that primer is made out of, and in humans, that's RNA. So an RNA primer is laid down. The reason that RNA primer is laid down is because the polymerase requires a 3' OH. That primer gives it, and it also requires double-stranded DNA, or in this case, double-stranded RNA hybridized to DNA. But it has it needs 2 strands, and then it can continue. It can copy, and it can only copy from 5' to 3'. So if it's copy, if I drew a copy in this direction, you should automatically know this is 5. That makes this 3. That makes this 5. That makes this 3. So you should be able to label all these ends from one end being labeled. And that means that this is the continuous strand, and this one is going to be made in fragments. So we got lots of primers laid down here. And I might ask you what are the name of those fragments. You tell me Okazaki fragments. All right, so once primase has done that, the DNA polymerase, remember you need to know that this is polymerase 3, copies the DNA. When it sees an A on one strand, and I will have this on the test, so I'm going to have something like this. And I'll ask you what's the other strand. So that if you didn't do that lab, I strongly suggest you do it to prep for the final. There's going to be several questions like that. So DNA polymerase 1 copies the DNA, and remember it always copies it in a 5' to 3' direction. So if that's 5, this is 3, and then we just go and match the letters. And this is also identical to me writing it like this. These are the same because this is just 3' on the left side, 5' on the right side. Those are the same. All right, DNA polymerase 3 copies the DNA, and then when it gets to the end, ligase has to fuse the ends together. And then the last thing is the RNA primer is removed by DNA polymerase 1. And that's replication. So make sure you know all that stuff. Remember that it's fast and accurate. I don't need you to know those exact numbers. I don't care. This is what we just talked about. Origin replication, helicase opens it up, single-stranded binding proteins hold it together. Here's the forks. Remember in prokaryotes, there is only one origin of replication. In eukaryotes, there are many. Otherwise, you'd never finish copying your DNA. You learned this in chapter 5, but just make sure you know that these are called nucleotides. Remember the DNA is anti-parallel, so 5' on one end, 3' on the other. This talks about the primase and DNA polymerase 3, so we already talked about that. And then DNA polymerase 1 removes that primer. We talked about one strand is leading, it's continuous because it's 5 to 3. That's the only direction that DNA can copy, because that 3' end has a hydroxyl where the 5' end has a phosphate. And remember from chapter 5, we talked about this, we have to do dehydration. And that means we have to take out water. You can't make water with the letter P. Well, everybody knows water is H2O, so you need H's and O's. And that's the only way that you can get it. So that's why DNA is always copied. Nucleotides are always added on the 3' end. And that's why this is copied backwards, because it goes in the same direction. Discontinuous is the ones with Okazaki fragments, so make sure you know continuous replication, discontinuous, the definition of that. Okazaki fragments, and then DNA ligase. Make sure you know all that. You should know this table, right? Priming is done by primase for both leading and lagging strand. Elongation is done by polymerase 3, specifically replacement of the primer is done by polymerase 1, and then joining of fragments is done by ligase. Remember, helicase and single-stranded binding proteins unwind the helix for both and keep it open. There's some videos I showed you that are in Canvas. We talked about the DNA replication. The leading strand always shortens because you can't add on the 5' end. So every time your DNA replicates, every time you go out in the sun and you kill skin cells, they have to make new skin cells, your DNA shortens. There's nothing you can do about it. The only thing that you can do about it is to turn on telomerase, but this causes a significant increase in your probability of developing cancer. And so a lot of people want to live forever because you essentially could. This causes apoptosis, this shortening. So most cells divide about 100 times. Cancer cells are immortal, so they can divide 10,000 times. Some people think by turning on telomerase that it will cause you to not age. That may be true, but you're probably going to die from cancer. So you look good, but you'll be dead from cancer. Telomerase is an RNA plus a protein, and it just basically adds on junk on the end. We talked about that. And I'll ask you that on the test. I'm not going to ask you the specific sequence of that. I don't really care that you know that. And it varies between organisms, so it's not really worth memorizing. I don't know what you're going to study. Eventually, you might study humans, or you might go to vet tech and study rabbits, and that would be different. The polymerase has a proofreading, right? And remember, we talked about like these thymine dimers. So know what causes thymine dimers. It's the ultraviolet light from sunlight. Know that those are covalent bonds, so they can't be fixed. They have to be cut out. Know that the enzyme that cuts it out is called a nuclease. The enzyme that replaces the cutout part is called DNA polymerase 1. Same one that replaced the RNA, and then ligase fills in the gap. So that's how DNA proofreading occurs and repair. And that's pretty much 16. Any questions about that? It's a lot of memorization for that chapter. Okay, so the last part for the test is chapter 17. So chapter 17, if you guys went through and did that assignment, and I've graded everyone's assignment already, so you should check it out to see. Anyway, the assignment is called Gene Expression Lab, and I think everybody should do that if they want to do well on the final exam. All right. So my advice to you is, if you haven't already done it, do the Gene Expression Lab. And if you don't know how to do the Gene Expression Lab, come to my office hours today after lecture or tomorrow, and I'll go through it with you. Also, to help you study for that, make sure that you do the genetics problems that I put on Canvas, and make sure you know how to get to the correct solution. If you have a problem with that, then I can also help you during office hours today and tomorrow. But this whole chapter is basically the central dogma, which is turning DNA, the A, G, C's, and T's in DNA into the A, G, C's, and U's of RNA, and then from that, turning that into the amino acids and proteins. So what you should know is that this first part, DNA to RNA, is called transcription. And RNA to protein is translation. And remember, these are two completely different things. Transcription is done by RNA polymerase. Sorry. So this is RNA polymerase. And this is done by ribosomes, which we talked about in chapter 6. Remember, in prokaryotes, the transcription and translation occur in the same place because they don't have a nucleus. In eukaryotes, transcription occurs in the nucleus, translation occurs in the cytoplasm. We talked about characteristics of a gene. So if I drew this out, I might ask you, what are the 3 parts of a gene and what's the order they're in? So promoters first, it's like the on-off switch or the dimmer switch. The coding region is actually the part that's turned into RNA. And then the terminator is what stops transcription. Promoters have different sequences. If you do that gene expression lab, it'll ask you to identify the TATA box and then all of the start codons after that. So that's good practice. I don't care that you memorize this stuff, but I want you to know that different promoters are different. They're kind of like a barcode, and that tells how strong it should be on. So how high should you set your dimmer switch? Should it be off? Should it be all the way up? Should it be halfway, 3/4, and so on? And that determines how much RNA is made. Transcription is done in 3 steps: initiation, elongation, and termination. So initiation is done by transcription factors. The TATA box we talked about, that promoter element, the DNA sequence, that's recognized by a transcription factor. The name of this is called TATA binding protein. You don't need to know that. But the reason I'm telling you that is because these things are real specific for the sequence that they bind to. And statistically, this is a relatively rare sequence. It would only occur because each of these letters is 1/4. We could do the statistics on it. It will be 1/4 to the 6th power, which is 8, 16, 32, 64 times 4 is 256. So this would only occur 1 out of 256 times. And if you're looking at a longer sequence, you know, we would go to 1024 and then 4096 and so on and so forth. So every letter you add in there is going to be much rarer. TATA binding protein finds this sequence and it binds to it. And that starts recruiting other transcription factors. Once they're bound to the other sequences, like the CAT box and the GC, they find their sequences and they bind. All these proteins recruit the polymerase. You don't need to know that it's 2, but it is. So an RNA polymerase is going to come in. It can read either strand, right? So it depends on what direction it's going. Since it's built this way and it's going from left to right, kind of like we read a sentence in English, it has to start at the 5' end and go to the 3' end, because remember, no polymerase can go to 3 to 5 because it can't add to that phosphate. It can only go 5 to 3 because it has to take water out. So that's why it would be this strand that's being copied. And so remember, the DNA sequence is going to be 3 to 5, which is anti-parallel to the mRNA, which is going to be 5 to 3. Together, we call this the transcription initiation complex. You should know that name, and you should know what transcription factors are. Once that's done, elongation is super easy. All it is that if the RNA polymerase sees a T on the DNA, right, then it's going to add a on the RNA. If it sees an A in the DNA, it'll add a U in the RNA, and so on and so forth. And then it continues to do that until it hits that terminator, which is actually a physical structure, like it's called a hairpin, which knocks the polymerase off. And that's termination. Once that's made, remember we modify the cap. So I'll ask you what goes on the 5' end of the pre-mRNA, and you're going to tell me a G, right? And I might ask you, what's the purpose of that? Well, it protects the RNA from getting broken down by enzymes in the cytoplasm. Those are also called nucleases. And then it also helps the ribosome know what direction to read this thing. The ribosome always reads the mRNA from 5 to 3 as well. So, and that's, we talked about that, you know, the words would be different if you read it backwards. The other modification is the 3' end. So I might ask you what's added to the 3' end, and you need to tell me it's a poly A tail, right? And that can, the longer it is, the more stable the RNA is. So the purpose of the tail is to inhibit degradation by enzymes in the cytoplasm, helps the ribosome know what direction to read, and regulates whether the RNA even leaves the nucleus, because without this, it won't leave, and it will never be turned into a protein because the ribosomes are only found in the cytoplasm. We talked about that before. The last thing that happens is called RNA splicing. Remember, we take out introns and snip and link together exons. This is just a combination dehydration and hydrolysis reaction. You're just cutting out the introns and then pasting the exons together. Make sure you know the term intron and exon. That's really important. And you can mix those up. So make you some flashcards to make sure that you know that. You should know that splicing is done by the spliceosome. You probably should know that the RNA is called small nuclear ribosomal RNA protein particles. SNRPs, we call them SNRPs for short. Just make sure you can pick it up because it's a multiple choice test. So know that the SNRPs are the RNA part of the spliceosome. They recognize sequences at the intron-exon junctions, which are here, right? So this is an exon. This would be like exon 1 in red. This is exon 2, and also in red, and this pink would be like intron 1. So it recognizes the junction between the red and the pink, right? The intron exon junctions, and it cuts and pastes. This gets tossed, and then now exon 1 and exon 2 are linked together. The purpose of this is so that we can make multiple RNAs from a single transcript. So we can shuffle it around. And we talked about those little words that you can put on your refrigerator, and you can move them around to make different sentences. That's kind of how it works, right? You can move these exons around to make different sentences that end up being different proteins. So in the beginning, scientists, before the human genome was done, we thought that we would need at least 100,000 or more genes in order to make something as complex as a human being. And everyone was shocked. There was kind of like some betting going on the side by nerdy scientists. You know, most bets were in the 100,000 to 200,000 range. I think the lowest bet was like 35,000 genes. And it turns out that it's even lower than that. It was like 20 to 25,000 genes. And the reason that they were wrong is because they thought one piece of DNA made one piece of RNA made one protein. So we knew that there were over 100,000 proteins. What we didn't know is this exon intron shuffling. And so it turns out, even though you might only have 20,000 genes, maybe every one of those genes can make 4, 5, 6 different proteins by flipping around the exons. And so you can have 100, 200,000 proteins and only have like 20,000 genes because of this splicing. All right. So that's transcription. Now put all of those note cards and everything in that one pile. The new pile is translation. And this is going from RNA to protein. This all occurs in the cytoplasm, different place, right? This is all done by ribosomes, not any polymerase. So make sure you keep all this straight. In the mRNA, there are codons. And again, if you guys do that gene expression lab, you'll know the codons are 3-letter codes. It's the only way we could get all the 20 different amino acids to be represented by using 3 letters. And I explained that in the lecture. And simply on the test, I will give you the genetic code and you'll just take the codon and turn it into an amino acid. Remember, it's always read 5 to 3. This table I will give you on the test, so you'll have that. You don't need to memorize it. It'd be like memorizing the periodic table. I think that's kind of dumb. I will ask you about the reading frame. Remember, the 1 shift or 2 shifts is really bad. 3 shifts aren't so bad because you go back into frame, right? And so I don't really need to go over this, but moving over, the ribosome moving over 1 letter instead of 3 will change every single amino acid, most likely. And that will wreck your protein. All right, so remember we have codons and we have anticodons. The anticodons are found on the transfer RNA or the tRNA, these little green dynamite sticks. Every TNA has a complementary 3 nucleotide sequence. So right here we have the anticodon, and this is the tRNA. The anticodon here is GGC. So that's going to pair with, CCG. This is going to be 3 prime. This is going to be 5 prime. So that's going to pair with GGC. C pairs with G. G pairs with C. Who's the one that figured that out? It's Shargoff. All right. So you need to know amino acid tRNA synthase. That's the enzyme that adds the amino acid to the tRNA. And the name tells you. Amino acid, tRNA synthase, it's making it. tRNAs plus amino acids. Every amino acid has its own tRNA synthase, right? Because every codon or anticodon would be a different shape. And you learned that in chapter 5. So there's 20 of these. Actually 64, but at least 20. All right, so you should know the E site, the P site, and the A site. You should know the A site is the one where the new tRNA that's charged, that has an amino acid attached to it, comes in. The P site is where the growing polypeptide is made, and then the E site is the exit where the uncharged, the tRNA with no amino acid, leaves. There's a video that we looked at for this as well. So just like transcription, translation has 3 stages: initiation, elongation, and termination. So initiation, you should know this. The small subunit scans till it finds the start codon. It's always AUG. So the anticodon is always UAC. The genetic code just gives you the codon. So the codon AUG should code for methionine. That's the first amino acid in every protein. And if you guys did that gene expression lab, you would see that. So AUG right here, you can see also it codes for methionine. It's right here. So this is the codon that you're going to be given for the test. So make sure you use the codons. Once that happens, then the large subunit is recruited, and then the next, whatever the next tRNA is that matches this codon, is going to come in here. So a little stick of dynamite with its amino acid on it. And it's going to come to the A site. This is going to get linked to this here. So it's going to cut and paste. It's a dehydration and a hydrolysis reaction. And then elongation is just the ribosome moves over 3 letters and it links it together and then moves over 3 letters and then links the next one and so on. So that the growing polypeptide is constantly changing positions. Here it's in the P site, right? It's going to move over 3 letters, so then it's going to end up in the A site right here. The one in the P site is going to end up in the E site, which is here. And then the one that is in the A site is going to end up in the P site, which is here. So it's kind of hard to see that on a flat screen, but watch some videos of how this works on YouTube. That'll kind of help you figure it out. And I also put some links to this in the pages in Canvas as well. The enzyme that does this transfer is called this transfer of amino acids from the growing chain to the new charged tRNA is called peptidyltransferase. You should know that term, so write it down, make a flashcard of it. I'll ask you that. Termination is when the stop codon is reached. So stop codons are UAG, UAA, and UGA. In genetics, I would make my students memorize that, but I'm going to give you this code, and it already tells you what the stop codons are right here. So you don't need to memorize those. When a stop codon is reached, there's no anticodon with the amino acid on it there. So it can't do the transfer. The release factor comes in with the anticodon for the codon, and it causes everything to disassemble. Now this protein goes off and folds in its correct shape. And we talked about this in chapter 5. The secondary structure and tertiary. And so you can go back to chapter 5 and take a look at that. But that's what's going to happen to this protein. It has to fold in the right shape so that it has the right function. All right, remember that an RNA that's being read by ribosomes is like a freeway with cars driving on it. You don't have to wait for a car to get off the freeway before you can get on it. You can share the freeway. And so lots of ribosomes, cars in this example, can be on one mRNA freeway, in this case, at the same time. So you can have multiple ribosomes on the same mRNA, and we call that polyribosomes. This occurs in both prokaryotes and eukaryotes. And that just means basically that you can make tons and tons and tons of protein off of one mRNA. You don't just make one and then you're done. All right, so after all this is said and done, the proteins have to fold in their correct shape. Most human diseases are caused by misfolded proteins, and that could be caused by mutations or all kinds of stuff. I mean, frankly, it's a miracle that we're alive. So the primary sequence of the protein, remember we talked about that, that's the order of the amino acids from amino to carboxyl. And you guys did some examples of this in chapter 5. After translation, the protein folds into its secondary and tertiary structures. We talked about chaperones. They can help fold it into that right shape. And then once it's all folded, we can change the shape even further by modifying it by these post-translational modifications. So after translation, we can add sugars, and that makes glycoproteins. We can add lipids, and that makes lipoproteins. We can add phosphates, and you guys know that this comes from ATP, and it changes shape. That's how we talked about this on all of chapter 8. And then we can modify the length of the amino acid chain, so we can take amino acid off. We can chop it in half to make 2 or more proteins. We can link them together to make coordinary structure. So all of that goes into the end product, which is what makes you what you are, flesh and blood. The last thing is in prokaryotes, remember, translation and transcription occur in the same place because there's no nucleus. So in prokaryotes, transcription and translation are coupled. In eukaryotes, transcription occurs only in the nucleus, translation only occurs in the cytoplasm. Prokaryotes, they're only a cytoplasm, so everything occurs in the cytoplasm. And termination is different, although I don't care if you know that or not. That's it. So do you guys have any questions about any of this? All right. Well, I'm not sure if anyone's there or not, but if you have any questions, hit me up in office hours. Good luck on your test. Make sure you read the announcements and stuff. I'll keep you up to date on any modifications or whatever. But for right now, the exam will be open. I think it's already open, maybe. I think I opened it on Saturday or Sunday. So you can take the test whenever you want, all the way up until the 23rd at midnight. It has to be done by then. So start early, and then I will calculate grades. I hope to be done by around noonish on Monday, and then I'll tell you in an announcement your grade, your final grades, and then I will give you an opportunity to take a W, or you can take the grade that you got. And then I have to have grades in by 5 o'clock on Monday. So like I said, I allowed students to go back and do the extra credit, but that's it. You can't go back and do labs and stuff like that. I just don't have time to grade everything. But you can do the extra credit if you skipped it.
With the fugate, the pedigree or whatever that was, that extra credit, how do you want that turned into?
You don't have to turn it in. The questions will actually be on the final exam itself.
Oh, okay. All right.
Any other questions? All right, good luck. It's been a pleasure.
Thank you very much.
Have a great rest of your summer.
Thank you too.
Thank you, you too.